Good afternoon, everybody. Sir, I'm starting my class here. Good afternoon, everybody. I hope my voice is audible to you. So, today we'll discuss the topics which were covered in week 7 lecture. So, week 7 is about snRNA, rRNA, miRNA, siRNA processing, export, and function. So, in this week, there are 6 lectures, and we'll very quickly and briefly summarize what all was discussed in these 6 lectures, and then we'll move to the questions. We'll try to do the questions simultaneously also, and we'll see that how we can mix the questions with the theory part. So, up till now, we have been studying about the many different aspects of RNA. Okay, so we have been looking at how the biogenesis happened, and how the life came, and how the evolution happened, and how the genetic material has shifted from RNA to DNA, and what is the RNA world hypothesis, which has essentially led to the evolution and to the creation of life. And then we have moved and learned about how the RNA is transcribed, what are the post-translational modifications that happens in RNA, what are the various alternative processings that happen in RNA, and how the regulation of RNA happens. So all these things we have covered. But we know that RNA is not just one kind of RNA. Okay, there are many different types of RNAs. Essentially, we study about mRNA, mRNA, rRNA, and tRNA. mRNA is the messenger RNA, which is made during the transcription process. And this mRNA, which is the messenger RNA, actually has the codons which codes for the proteins. So whatever information that which protein has to be formed, that information is present in the form of codons in mRNA. rRNA is the ribosomal RNA which is involved in translation process, and tRNA is the transfer RNA which is also involved during the translation process. It brings the charged amino acid. But these are not the only kind of RNAs which are present. There are many different kinds of RNAs as well. So these RNAs, which are known as non-coding RNA, which comprises of snRNA, rRNA, miRNA, siRNA. So your rRNA and tRNA are also your non-coding RNA. Why? Because coding RNAs are the RNAs that codes for a protein. Okay. So any RNA that codes for a protein is your coding RNA. And coding RNA is your mRNA. Because mRNA only codes for the protein. mRNA has the codon information which codes for the protein. Rest rRNA, rRNA, and tRNA, they help in the translation process, but they do not code for proteins. Okay. And then these small nucleolar RNA, microRNA, siRNA, and many other different kinds of RNAs, these are known as non-coding RNA. So non-coding RNA are the RNA which does not code for a protein. Okay. Non-coding RNA do not code for a protein, but these non-coding RNA has other different regulatory roles. Which roles? Regulatory roles. So these non-coding RNAs have regulatory roles, and we have studied in the previous classes that they define the expressivity, that how much expression of a particular RNA will happen. These non-coding RNAs will define that. So they decides or define the expressivity of the genes. Okay. So earlier these non-coding RNAs were like, we didn't know what is the function of these RNA, and we thought that they do not have any function. But now we know that these non-coding RNAs are indeed very much important, as much as they are as much important as the mRNA. Okay. So this week we'll be covering that how these snRNA, these rRNA, miRNA, siRNA are processed, exported, what are the functions, and what diseases can occur if the function of these non-coding RNA is altered. Okay, if the function is altered. So what can happen? What are the diseased conditions that can happen if the function is altered? Okay. So the first lecture in this series is siRNA, rRNA, miRNA, siRNA processing, export function, introns, and link to splicing. So there are various mechanisms by which introns are gained, and mostly these non-coding RNAs are present in the intronic regions. That's why we are studying about the mechanisms of intron gain or loss. Okay. So there are different mechanisms by which uh, introns can be gained. So let us see what are those mechanisms. It can be intron transposition or transposon insertion. So first of all, intron transposition. So what is intron transposition? Let us see. This is these are the this is one G. This is the intron. Now these are exon 1, exon 2, and in between there is intron. So the transcription happens, and then during the post-transcriptional modification, this intron is spliced out. So you can see this intron is spliced. Now this spliced intron can go and it can reinsert into any other site. Okay, so you see spliced intron reinserts into mRNA of two genes at an intron-less site. Intron-less site means, suppose this is a gene and it does not have any intron. So there are no sites of introns. So this spliced intron go and it joins in between this particular gene. See, there were no intron. It was a complete one exon. Here, this intron comes and binds. Then reverse transcription of RNA of gene 2 with new. Now then reverse transcription happens and now it's a complete new gene which has been formed. So this is intron transposition. That means intron changes its position from one exon to another exon. From one exon, when it is spliced out, it goes and it binds to any other gene at an intron-less site. This is important. This is your intron transposition. Then comes your transposon insertion. So transposons are also known as jumping genes. So see, this is a transposon. It can go and it can insert itself and then conversion to spliceosome intron, and then this transposome is converted into a spliceosomal intron. So transposomes have inverted repeats at both the ends and have a single enzyme called transposase. So because they have inverted repeats and they have an enzyme which is called as transposase, so they can just go and they can leave a place and they can bind to somewhere else. So if you study in detail about these transposomes, you will get to know that they can insert itself by two mechanisms. There are two mechanisms by which transposomes can be inserted. The first mechanism is the cut and paste mechanism, and the other one is the copy and paste mechanism. Cut and paste means that transposome is just taken out from one place and that particular transposome is only inserted to other place. Copy in place means that this transposome will first make its copy, and now this copy will go and bind to somewhere, not this intron transposome only. So there are cut and paste and copy and paste. Two types of mechanisms are there, but these are the two mechanisms of intron gain. That how intron is gained by a cell, by a gene. One intron which is spliced which goes to another gene, or transposome can be converted to an intron. It is converted to a spliceosomal intron. Okay, I hope this is clear. So now there are certain two terms which needs a lot of discussion. That's why I've included it here. Just a minute. Okay. So this is homology. Homology, which means This is important to understand. What are homology means that they have a common origin point. They have a common ancestry. So whenever we talk about homology, we talk about a common ancestry. That is, they have a common point. So homologous genes become separated in evolution in two different ways. So suppose these are the genes and they have a common origin. Now they can be separated into two different ways. Okay. Now what are these two different ways? First is separation of population with the ancestral gene into two species. Okay. First, it can either separate into and they can form two different species altogether. This is two different species, or gene duplication of this gene within a lineage, or they can have within this lineage, they can have a gene duplication event. Anything can happen. Either they can form two different species or they can have a gene duplication event. When genes are separated and they form two different species, in that case, speciation is said to have occurred. Speciation. And here, gene duplication. So genes which are separated by this speciation event and form two different species, these genes are known as orthologs. And genes which are separated by gene duplication events, these are known as paralogs. So orthologous and paralogous are the two branches of homology only. So orthologs are the genes which are separated by gene speciation events, and paralogous are the genes which are separated by gene duplication events. Okay, fine. So these are the two terms which are important to understand when we talk about homology. So homology means common ancestry. In homology, you have two branches, orthologous and paralogs. Orthologs are the genes which are separated by speciation events, and paralogs are the genes which are separated by gene duplication events. Okay. Next is the regulation of RNA splicing. We have talked about RNA splicing, that RNA splicing itself is a post-transcriptional regulation. That means it controls that whether the transcription has happened properly, and it will remove the introns and then will make the uh, naive RNA into a mature RNA or pre-RNA into a mature RNA. But this splicing event is also regulated. Okay. So post-translational and protein modification. So how this happens? This RNA splicing is governed by two kind of spliceosome complex. This we have already studied in the previous class also. There is one major spliceosome and the other one is a minor spliceosome. So major spliceosome has U2 type introns. Okay. Major spliceosome, it has U2 type intron, and minor spliceosome, it has U12 type of intron. So the major type of intron is the major spliceosome. And you can see here in U2 type of splicing, the sites are AG and AU, AG. यहाँ पे AG and CG. So this is the difference. Then what are the post-translational protein modifications? So after the translation has happened, what forms? After translation happens, protein is formed. But that particular protein also undergoes some kinds of protein modifications. Okay. So there are not only post-transcription modifications that happens, there are post-translational modifications also that happens. So what are the post-translational modifications that happen in the protein? Post-transcriptional modifications happens in RNA, mRNA. RNA. And post-translational modifications, they happen in proteins. So post-translation modifications can be phosphorylation, acetylation, hydroxylation, and ubiquitination. These are the post-translational protein modifications, PTM. Okay. Phosphorylation means addition of the phosphate group. Kinases, okay, acetylation means acetyl group is added. Ubiquitination means when they are ubiquitinated, they are marked for degradation. If the protein is non-functional, if the protein which has formed is non-functional, there are some problems in the proteins, then that particular protein is ubiquitinated and then it is marked for degradation. Okay, fine. Then these are some of the pathologies that results from splicing defects. So you understand that it looks like that splicing is such a small phenomena that happens during the post-transcriptional modifications. But each and every biological process that happens inside the cell, that happens inside the body is very carefully and articulately regulated. And if there is any problem and any defect that happens, it can lead to some kinds of diseased conditions, or it can lead to some kinds of difficulties or chronic malignancy also in some cases. So here, these are the mutations. So you can see that if there is any mutation and that affects the splicing factors. So there are two things. Mutation can happen that can affect your splicing factors, or mutation can happen that can affect the splicing pattern of the pre-mRNAs. Anything can happen. Either the splicing factors are affected or the splicing pattern of the pre-mRNAs is affected. So if there is any effect on the splicing factors, these are the conditions that can happen. Spinal muscular atrophy, retinitis pigmentosa, and myotonic dystrophy. So these are the diseased conditions that can happen if there is any effect in the splicing factors. And if there is any effect in the splicing pattern of the pre-mRNAs, then you can have beta thalassemia, you can have muscular dystrophy, you can have cystic fibrosis, Frazier syndrome, or you can have frontotemporal dementia and Parkinson disease also. Okay, so just to tell that each and every, even a small molecular biology that we study, a small process, even a small, very small biological process can have a greater or a larger impact. Then we come to lecture 2 where we talk about the RNA helicase. So helicase essentially unwinds the nucleotide, whether it is DNA or RNA, it unwinds it. Helicases perform the action of unwinding. Whether they are DNA helicase, they will unwind the DNA. If they are RNA helicase, they will unwind RNA. So if we talk about RNA helicase, these are RNA dependent NTPase or RNA unwindases. So what they do is that they hydrolyze the NTP. They hydrolyze the NTP and then therefore they unwinds your RNA. So since they are RNA helicase, what is their substrate? Their substrate can be either your double-stranded RNA or it can be your RNA protein complexes. Anything can be your substrate. If if the substrate is double-stranded RNA, it will only affect on double-stranded RNA. If the substrate is RNA protein complex, then it will affect the RNA portion of the RNA protein complex. And this is the RNA helicase family. So you can see RNA helicase family comprises of ATPase, helicases. I mean, these are just this is the sequence of how it happens. Okay. Next we come to a little bit more about the RNA helicases. So these RNA helicase, for its activity, it requires 7 conserved motifs. Okay. So 7 conserved motifs are required for the activity of RNA helicases, and the N and C terminal extension for substrate specificity. If you see here, the N terminal or the C terminal of ends of the RNA helicase, they are required for the substrate specificity. Okay, this is important. So substrate specificity is determined by the N terminus as well as the C terminus. Okay. Then RNA helicase in ribosome biogenesis. So these RNA helicases are also involved in the biogenesis of ribosomes. So how they help? Because they help in the snRNA pre-RNA base pairing. They help in RNA protein interactions, where these are the places where they act. snRNA, small nuclear RNA, pre-RNA base pairing on the during the activity of endo and exonucleases, intra and inter-RNA RNA interactions, or RNA protein interactions. All these places, these act. Now, you can see processes requiring RNA helicases. So we know that in DNA, RNA helicase are required during which process? It is required during DNA replication. So when we have DNA replication, there we have studied that DNA helicases are needed. Okay. Now here we are talking about, okay, we know that there are RNA helicases, but what is the use of these RNA helicases? Where these RNA helicases are needed? That is also one thing that we should know, that where these RNA helicases are needed. So these RNA helicases are required during pre-mRNA splicing, during RNA maturation, during RNA export, during the translation process. So you can see that these RNA helicases have multiple roles. They are required at so many levels. They are required for the splicing, they are required for the maturation of RNA, they are required for the export of RNA, they are required for the translation events to happen. So there are multiple roles of these RNA helicases. And more than 8 RNA helicase participate at various steps of RNA splicing, and helicase helps in unwinding of RNA or RNA protein complexes. The major role of helicase is that it will unwind. So wherever there is RNA bonds are there, RNA complexes are there. So these helicase helps to unwind those RNA complexes, or they can also help in unwinding the RNA protein complexes. So here in translation, you have RNA protein complex. Even here, see, transcription factors are also proteins. So if any kind of unwinding that has to happen between the transcription factors or the mRNAs or the RNAs, then there also RNA helicase will play the role. Okay. Next is RNA helicase in RNA export. See, here we were seeing that they are involved in RNA export also. So how RNA helicases are required in RNA export? Let us see that. So correct conformation of RNA protein complex is needed for the export through the NPC. NPC stands for nuclear pore complex. It is. So what does it mean? That correct conformation of RNA protein complex for export through nuclear pore complex. Unpacking of RNA protein complex in the cytoplasm, removal of shuttling proteins for their rapid transport to the nucleus, coupling RNA export to translation. So what happens is that when RNA is coming from the nucleus to the cytoplasm, some kinds of transporter proteins helps in the export of the RNA from the nucleus to the cytoplasm. Okay, there are certain transporter proteins which helps in the transport of those proteins, those RNA from the nucleus to the cytoplasm. Suppose this is your nucleus and this is your cytoplasm. If I take this area and expand it, so there are certain transporter proteins that combines with your RNA and helps in its movement outside the nucleus. Now these transporter proteins are bound to the RNA. They should be immediately and very quickly should be detached from the RNA so that they can go for another round of export by binding to another RNA. That means the detachment of the RNA with the protein has to be real quick and efficient and correct also, so that this motor protein can again go inside the nucleus, bind with another RNA molecule and help in its transport to the cytoplasm. Okay, so this unpacking of RNA protein complex is done with the help of RNA helicase. So these RNA helicase helps in the removal of these shuttling proteins for their rapid transport to the nucleus. Okay, so therefore these RNA helicases are important here. Here we must see that they are also involved in your RNA export. So how they help in RNA export is they help in they free the your RNA protein complex. So the protein is free, the transporter protein is again free to move inside the nucleus and perform its activity again. So in this way, these RNA helicases are very important in the case of RNA export. Now, what is their role in translation process? Here we saw that they are also involved in translation process. So how they are involved in translation process? So intra-RNA interaction in mRNA, ribosome assembly and disassembly, and translation termination. So these are the three events. Okay. These are the three events in which RNA helicases are required during the translation procedure. Okay. Which means that intra-RNA interaction in mRNA. Suppose this is your RNA. Because of some kind of base complementarity, this portion of RNA now starts interacting with this portion of RNA and it will form some kind of loop structures. So this kind of loop structures is a problem for the cell. So this kind of intra-RNA interaction can be prevented with the help of RNA helicases. Then it also helps in the assembly and the disassembly of the ribosomes, which attaches to the RNA. And in the termination of translation also, these RNA helicases helps because it can help in the detachment of the proteins, the raw proteins that has bound to the RUT sequence. So it can help there also. But that is in termination. Okay, we're talking about termination. That is in translation, but here also it helps in the detachment process. Then we come to the nuclear pore complex. Nuclear pore complex helps in the import and the export of the proteins and the RNA. So there are two things, two phenomena on two processes that happens with the help of these nuclear pore complex. Okay. The one is the nuclear export and the other one is the nuclear import. Okay. Export means it is going out of the nucleus. Okay. Here in this context, and nuclear import going inside the nucleus. Is it clear? Just read these. I'll give you 2 minutes. Just read all the differences or the comparisons which are given. See here, nuclear export require the NES signal, which is the nuclear export signal, and nuclear localization signal is required for the nuclear import. Just read all these things. Just a minute. I think there is some technical error. Just wait for 1 minute. अच्छा यार, मैं ना अपनी मम्मी से पूछती हूँ कब करवाना है। वैसे तो वो मंडे बोल रही थी, क्योंकि मंडे सोमवार अच्छा रहता है दिन वैसे तो मैं एक बार पूछती हूँ मम्मी से, तो फिर बताती हूँ। थैंक यू। ओके, so I hope you have read all these comparisons. So for nuclear export, your nuclear export signal is needed, whereas for nuclear import, the nuclear localization signal is required. Now, the transport receptors are known as exportins. Here, the transport receptors are known as importins. For both the exportin and importins, what is needed? Small GTPAs are needed. But here, these small GTPAs are known as RAN. And this RAN, here in the case of export, are known as RAN GTP, and here it is known as RAN GTP. Then, this is the export cargo complex. Here it is known as import cargo complex. And these are the differences. You should just read about it. Then we come to the nuclear cytoplasmic transport. How this nuclear cytoplasmic transport happens. So, this is an important image. So, you see, this image specifically shows the movement of cargo proteins or RNA through the nuclear pore complex. Here we'll consider the movement of the RNA through the nuclear pore complex. Okay. So, what are the key components of this image? Key components of this image are RAN GTP, the green triangle one that you can see. This RAN GTP is essential for the directionality in the transport. Okay, this defines the directionality in the transport. Then we have transport receptor, the blue structure that you see. This helps in cargo recognition and transport. Then cargo can be, here you see cargo can be anything which is going inside and outside of the nucleus. So here we can say cargo is the RNA. The molecule which is transported is your cargo. I'll just write over here. Cargo is the molecule which is transported. Then FG repeat domain, the yellow circles that you see, these yellow circles everywhere, these FG repeat domains are found in nucleoporins and they provide a selective barrier for the transport. Okay, these FG repeat domains They provide a selective barrier for the transport to happen. Then you have zinc finger domain and amino terminal domain, and they are likely related to cargo binding and interaction with the transport machinery. Now, if we look at the process overview, so what happens is your RNA molecule. Okay, here in this case, your cargo is your RNA. So this RNA bind to the transport receptor in the nucleus. Okay, see, this is the transport receptor. It binds to it. That complex moves to the nuclear pore complex. So first you see, this is the nucleus and this is the cytoplasm. Okay, this is, it goes, the transport receptor and the RNA, it goes and it binds to the FG repeat domain. Then it goes, this is opened and it goes inside. Then what happens? RAN GTP hydrolysis happens, which causes the release of the cargo. The empty receptor is then recycled back into the nucleus. Okay. So this system, it ensures the controlled movement of proteins and RNA. Therefore, it maintains the cellular function. Important thing to understand is that it has the RNA molecule along with the transporter receptor. It has to bind to the FG repeat domain. Then only it can go and it can uh, go inside and it can function. Okay. Now we'll come to questions. So what we'll do is that we have completed up till uh, lecture 2 we have completed. We have a lot to go. So we'll do one or two questions and then we'll come to the lecture 3. So the first question is, FG nucleoporins are located in, and the options are A, nuclear membrane, B, nucleolus, C, nuclear pore complex, and D, cytoplasm. Okay, I'll give, after 2 minutes, I'll switch the screen. Right now it is 12:35. I'll switch it 12:37. See, you have 1 more minute left. Okay, now only 1 minute is left. Okay. So The correct answer of this question is nuclear pore complex. Okay. So the question is Fg nucleoporins are located where? So Fg nucleoporins are located in the nuclear pore complex. So these proteins, basically these Fg proteins contain phenylalanine and glycine repeats. Okay. So if we talk about these Fg proteins, which we will, I think we will eventually talk about, they have phenylalanine and glycine repeats. Okay. And therefore, they play a crucial role in the nucleocytoplasmic transport. How? Because they form a selective barrier within the nuclear pore complex. So these Fg proteins basically forms a selective barrier. Just like you have a semi-permeable membrane, here this is Fg proteins which forms a selective barrier. So therefore, the correct answer is nuclear pore complex because they are helping in the transport. Next question is exportin T function depends on A. RNA quantity inside nucleus, B. RAN GTP, C. RAN GAP, and D is exportin 5. Okay, 2 minutes to answer this question. Right now it is 11:38. I will switch the screen at 12:40. One more minute left. Okay, now I'm switching the screen. So the correct answer is RAN GTP. So how? Let us see. Exportin T is responsible for exporting tRNA from the nucleus to the cytoplasm. Okay. This exportin T, what is the function of this exportin T? It is responsible for the export of tRNA from the nucleus to the cytoplasm. Okay. And in this process, it requires RAN GTP. So this RAN GTP, it forms a gradient across the nuclear membrane, which facilitates the transport of cargo molecules through the nuclear pore complex. Is it clear? So the correct answer would be RAN GTP. Okay. Now we come to lecture 3. Here we are again talking about more about the RNA helicase. So you see how it happens. This is the nucleoplasm. This is the cytoplasm. So this is your RAN GTP. This RAN GTP, it goes to the cytoplasm. Here it is converted to RAN GTP, and then this RAN GTP comes from the cytoplasm to the nucleus. Okay, and then it gets this GTP, and then it gains one ATP molecule, and then again it is converted to GTP. So GTP has to go from the nucleus to the cytoplasm. Okay. GTP can go from the nucleus to the cytoplasm, and GDP can come from the cytoplasm to the nucleus. GDP. It can come from the cytoplasm to the nucleus. So, the different RNA species are exported through the nuclear pore complex via mobile export receptors that we have discussed. Small RNA follow relative simple export routes by binding directly to the export receptor. So, these are the export receptors to which your RNA molecule comes and bind, and then they move along the nuclear pore complex, and then they shift it from the nucleus to the cytoplasm or vice versa. So if you have a small molecule, a small RNA molecule, okay, so if you have a small RNA molecule, this small RNA molecule can go from the nucleus to the cytoplasm simply by binding to these mobile exporters, and nothing else is needed. But if we have large RNA, small RNAs can be your tRNA and microRNA. But if you have large RNA molecules such as ribosomal RNA or mRNA, then they have to assemble into a complex. And this complex is known as ribonucleoproteins. Okay. So if you have small molecule, this is important. If you have small molecules like small RNAs, then they have a simple, relatively simple export routes. They can directly bind to the export receptors and they can go. But if you have large RNA, then they assemble into ribonucleoprotein particles and then recruit their exporters via specific adapter protein. So first they form RNP, that is ribonucleoprotein complexes, and then they are recruited to their exporters by binding to the adapter proteins. Export of mRNA is unique as it is extensively coupled to transcription and splicing. That is fine. Now we come to lecture 4, where they talk about nucleoporins in RNA export. That how these nucleoproteins helps in the RNA export. So we have already talked about that these nucleoproteins are made up of nuclear pore complexes are made up of pore proteins, and these nucleoporins are of three major classes. This is important. Nucleoporins, when we talk about nucleoporins, these nucleoporins have three major classes. First is FG nucleoporins. Second, nucleoporins which do not have FG. So, no FG nucleoporins. And then you have third class which are known as NUPs. NUPS, NUPs. Okay. So first class is FG nucleoporins, which contain this repeat, phenylalanine and glycine repeat that we have just talked about, that fill up the active transport channel and functional directly in the nucleocytoplasmic transport by mediating the passage of the soluble transport receptors. Second class is nucleoporins that do not have this FG proteins, and they are predominant structural components of the nuclear pore complexes. Majorly, they are present. And the third is the NUPs. and which are integral membrane proteins and are thought to anchor the nuclear pore complex in the nuclear membrane. So the nuclear pore complex is attached to the nuclear membrane with the help of this NUPS. Okay. Now we come to tRNA export. How the tRNA export happens. So for the tRNA export also same principle is there. The tRNA export happens with the help of exportin T, which is a member of just hmm. this tRNA export happens with the help of exportin T, which is a member of the karyophyte superfamily, and the nuclear export structures they are. So this is nothing. This is just read. I have just included the screenshot so that you can go and read about this. Nothing much to explain. These are all the self-explanatory sentences just for you to revise and recapitulate. I have added. Important point here is that exportin T is needed for tRNA transport. Okay. I'll just write it over here that exportin T is needed for tRNA transport. Next is biogenesis and export of miRNA. So how miRNA has forms and how then miRNA is exported. So now this is important. The biogenesis of mRNA is important, how mRNA has evolved and how mRNA is formed. So I'll explain you this with the help of this figure which is given over here. Okay, because then it becomes easier. So let me explain this figure to you. See, this is your, just do it like this. This is your primary mRNA, miRNA, pri. So I will write it over here. Pri miRNA means primary miRNA. So this primary miRNA or pri miRNA is used as a template, is transcribed, okay, with the template as miRNA gene. So what you have to understand is that you have this miRNA gene and this miRNA gene is transcribed into this pri miRNA, which is primary miRNA. Now, on this primary miRNA, DROSHA and DGCR8, DROSHA and DGCR8 act and it cleaves the primary miRNA. After the cleavage, it forms pre-miRNA. So you understand? So what is happening? First is your pri-miRNA. This pri-miRNA is formed by the transcription from the miRNA gene. Now on this pri-miRNA, DROSHA and DGCR8 acts and it cleaves the primary mRNA and it cleaves the primary miRNA and then it forms the pre-mRNA, which is the precursor miRNA. Here, it forms pre-miRNA, which is the precursor miRNA. Okay. Now this precursor miRNA will go from the nucleus to the cytosol with the help of exportin 5 transporter protein. You see here, this is exportin 5 here. This exportin 5 go, and this pre-mRNA binds with this exportin 5. And then this complex from the nucleus is transported to the cytosol. So, exportin 5 it transports the pre-mRNA from the nucleus to the cytosol. From the nucleus then what happens? Now it comes, now this pre-mRNA is there in the cytosol. Now the maturation of this miRNA, it happens in the cytoplasm, in the cytosol. How it happens? Dicer enzyme comes with the help of FMRP. This FMRP is fragile X mental retardation protein. So this dicer comes and it cleaves the pre-miRNA into miRNA duplex. So dicer acts in the cytosol and drosha acts in the nucleus. This is important to remember. The dicer, it acts in the cytosol, and drosha, it acts in the nucleus. So what happens? Then this dicer cleaves, dicer comes and it cleaves the pre-mRNA into miRNA duplex. So then your miRNA duplex is formed. Now this miRNA duplex has two strands. Okay, what happens? One of the strand, it goes and it binds, it is incorporated into this risk complex, which is RNA induced silencing complex, and the other strand, which is the passenger strand, is degraded. Okay, so one of the strand, it goes into the risk complex, and then when it goes to the risk complex, which is RNA induced silencing complex, this is RNA induced silencing complex. Now, when the this miRNA is incorporated into the risk complex, then it can perform its function. Okay. And the function can be translational repression or mRNA cleavage. What can be the function? Function can either be translational repression or it can be the cleavage of mRNA. So translational repression means that mRNA is formed, but the translation process is replaced. So there would not be any translational process, but mRNA has formed. And mRNA cleavage means that mRNA itself, at the mRNA level only, the mRNA is cleaved. So there is no possibility of going up till translation process. Here, mRNA is not cleaved, but the translation process is repressed. So they both are two different mechanisms by which this miRNA can function. So now if you look here, what happens? Biogenesis and export of miRNA. The stem loop structure is cut. Here is the cursor. The stem loop structure. The stem loop is cut in the nucleus by drosha in complex with DGR8. And then this pre-miRNA is exported via exportin 5 and then it is cleaved by dicer. So drosha acts in the nucleus, and dicer acts in the cytoplasm, and the protein transporter protein which is responsible here is exportin 5. So I hope this miRNA biogenesis and its export to the cytoplasm is clear. So the biogenesis step itself involves the export. That means the biogenesis process is complete only when the export happens. Okay. So now we'll just solve 2, 3 questions and then we'll go to lecture 5 and 6. So in this week, we have a lot of lectures. We had 6 lectures. So that's why we are trying to cover all the 6 lectures, although we cannot go in detail about each and every lecture because if we start doing that, we won't be able to do the questions and the theory simultaneously. So we are just looking at the important topics from each and every lecture. Okay. So let us see these 2 questions and then we'll move further. So the first question, the next question is, length of poly A on mRNA tail indicates that mRNA is undergoing decay. mRNA is ready for translation, age of the mRNA or nuclear location. So what does the length of poly A tail on the mRNA indicates? Okay, 2 minutes and then I'll switch the screen. Right now it is 12:58. I will switch at 1 o'clock. Okay. One more minute left. Okay. Okay. I'm switching the screen now. So the correct answer is age of the mRNA. So length of the poly A on the mRNA tail indicates the age of the mRNA. Okay. So how it happens? Because the newly synthesized mRNA have a long poly A tail of about 200 to 250 nucleotides, which stabilizes the mRNA and promotes translation. As the mRNA ages, the poly A tail shortens due to exonuclease activity, and eventually it leads to mRNA decay. So, a very short or absent poly A tail signals that the mRNA is nearing degradation. So, the length of poly A tail serves as an indicator of mRNA stability and age. So, always remember that newly synthesized mRNA, when you have a newly synthesized mRNA, so this newly synthesized mRNA have a long poly A tail. Okay. Newly synthesized mRNA have a long poly A tail of about 200 to 250 nucleotides. Because this long poly A tail, what is the function of poly A tail? It stabilizes your mRNA. But then eventually, as the mRNA is there, exonuclease activity comes and it keeps on degrading to the mRNA, which keeps on degrading the poly A tail, ultimately which can lead to mRNA decay. So the length of the poly A tail indicates the age of the mRNA. This is the correct answer. Next question is, miRNA maturation is dependent on DROSHA, DGCR8, dicer, or risk. This we have just now studied in detail. Okay, now I'm switching the screen because this is an easy question only. The correct answer is dicer. We have just discussed that maturation of the miRNA, maturation of the miRNA, this happens in the cytoplasm. And in cytoplasm, dicer only acts. So, dicer is the correct answer. Okay. Next, we will come to lecture 5. So, lecture 5 is about the RNA export mechanisms ka continuation. So, this is all like, it's not very much to explain. These are all self-explanatory that how the export of snRNA happens. You have to just read it. I have included it so that you can read it. What thing you have to understand is that the proteins which are involved, for example, exporting one is involved, and adapter protein, which is FAX, and then the phosphorylation of the FAX protein that happens. So these are the important points, that how, what are the what is the transported protein, and what are the adapter binding protein, and what are the modifications that happens in the transporter protein. Okay. Then next is the same, the transport of snRNA. So snRNA transport, you have SNM complex, which is important for mRNA. You have the NUPS proteins, which are important. So this is all just read. You can just read through it. Not much to explain here. Then you have transport of mRNA. Then different transports. No need to go into very much detail. Just you just need to know some names. Okay, some names are important. That much only you need to know. No need of going into much detail into it. Okay. Now this is lecture 5, the descriptive part, like whatever the theory part is there, just go through once and just try to read and as much as you can remember. No need of that much depth. But yeah, then this is important for your competitive examination points of view. Then what are the diseases that can be linked to it? So, defects in the mRNA nuclear export pathway can have direct consequences on human health and development. How? So, There are some points for it. Hijacking of the endogenous mRNA machinery by viruses. Then what happens that fragile X, this FMRP, the protein that we have just studied when we were studying about the export cycle. So what happens that it can destabilize. FMRP interacts with NXF1 homolog, NXF2, and it destabilizes, and it can lead to a disease which is known as osteogenesis imperfecta. And this disease is associated with the aberrant mRNA transport. What happens? A specific splice site mutation happens in one cohort of patient, it has been reported, which lead to defects in splicing and nuclear retention of the collagen mRNA. So this is the disease osteogenesis imperfecta. This is important. Osteogenesis imperfecta. This disease happens when there is some destabilization that happens in the FMRP protein. FMRP protein. It destabilizes and then there is aberrant mRNA export happens. Which leads to osteogenesis imperfecta. Okay. So now we come to RNA decay. We have studied what happens RNA decay. Most studies of RNA decay are done with mRNA. Cells need to improve mRNA that are damaged or not needed anymore. mRNA decay pathways are found in both nucleus and cytoplasm. So mRNA have two stability determinants. So this is your mRNA. How this mRNA can be decayed? There are two regions through which mRNA can be decayed. How? By knowing the stability determinants of mRNA. What are the stability determinants of RNA? So first is the 5' end and there is a 3' end. Okay, so your RNA has a 5' end and RNA has a 3' end. So the attack of exonuclease, it can happen either from the 5' end or it can happen from the 3' end. So these are the two areas or the regions in the mRNA which are open, which are more prone to attack by exonuclease. So these are the two stability determinants points of an mRNA. And these 2 points are covered with a 5' cap and a poly A tail. The decay of the mRNA can happen in 3 ways. What are those 3 ways? First is there is some attack from the 5' end. There is some exonuclease attack obviously, and there is some exonuclease attack from the 3' end, or there is any endonucleatic attack in between. If there is any endonucleatic attack in between, that can also lead to decay. So 3 points of decay, 3 points of action, 1, 2, and 3. But you have a 5' cap here and you have a 3' cap here. So a 3' poly A tail here. So as long as your 5' capping and 3' tailing is there, it will ensure that there is no attack that's happening from either of the ends. Okay. But as the RNA ages, we have just studied that as the RNA ages, the poly A tail gets attacked by endonuclease, it gets removed. Eventually, the poly A tail is completely removed and then the RNA becomes very much prone to decay. So this is your RNA decay. Decay of the RNA can happen either from the 5' or the 3' end or from the endonucleatic attack. Major decay happen from the 5' or from the 3' ends. Okay. And here the poly A tail, as the decay happens, the poly A tail gets shortened and shortened and your mRNA gets even more prone to decay. Now we talk about the RNA quality control. So when we talk about RNA quality control, transcription quality control. So these are the various checkpoints. So at each and every checkpoint, it is ensured that the quality of the RNA is maintained. And anywhere if they find, just a minute, so this is what happens. Some network, there is some internal. Hmm. So these are various checkpoints. So at each checkpoint, it has to be ensured that the quality of the RNA is perfect. Because if there is any compromise in the quality, now, what is the meaning of quality? Quality here means that the sequence is retained, the conformation is retained, the placement is there, the export and imports are perfect. So these are the quality controls. So during the transcription, quality control is there. If it is not, then it is degraded. During the time of capping, also quality control is checked, otherwise it is degraded. Splicing, polyadenylation, export to the cytoplasm, translation, and if not, degradation. This is the ultimate. This is the lifespan of our RNA. It will be transcribed, it will undergo the post-transcription modifications, it will be exported to the cytoplasm, it will perform its function of translation, and then eventually it will be degradation. This is the life cycle of RNA, and at each point, if there is any problem, then it can be, it is, first they will try to repair it. If there is a small problem or a short problem which can be repaired, it will try to repair it, otherwise it will go for degradation, so that there are no mutations or no uh, disease conditions can happen. So our body tries best to control any kind of abnormalities, abnormal conditions. So that is why this is the RNA control. Okay, now we'll come to the questions. A disease that is the result if defective mRNA transport happens. What are the options? Osteomalacia, osteoporosis, osteomyelitis, or osteogenesis imperfecta. So what is the disease which is a result of defective mRNA transport? Right now it is 114. I will switch at 180. 18. This question we have just discussed, so it should not be very difficult for you to answer this question. Okay, so now I am switching the screen. Hmm. The correct answer is osteogenesis imperfecta. So here only we have studied this, that if there is any defect in FMRP, the FMRP it gets destabilized and there is aberrant mRNA export that happens, and this is known as osteogenesis imperfecta. Okay. Next question is, hmm, RNA undergoes decay because A, it lacks coding sequence, B, it has stop codon, C, it lacks essential stability features, or it has intro. So 2 minutes to answer this question. Right now it is 116. I will switch at 180. One more minute left. Okay, so I'm switching the screen now. 2 minutes are over. So the question is, RNA undergoes decay because it lacks coding sequence, it has stop codon, it lacks essential stability features, or it has intron. And the correct answer is it lacks essential stability features. So we have already discussed this one. What are the stability features that are present in an RNA which helps or which prevents it from decay? We have discussed it has a 5' end, it has a 3' end. 5' end has 5' capping, and here you have 3' poly A tail. So poly A tailing is there. And these are the two stability features which prevents the mRNA or RNA from the decay. And as the RNA ages, these stability features also starts getting attacked by endonucleases, and therefore RNA gets more and more prone to decay with age. So now we have some true false. Nucleoporins are important in RNA transport. True or false? Sorry, important in RNA transport. True or false? 2 minutes. Right now it is 120. I'm going to switch the screen at 122. One more minute left. Okay, now I'm switching the screen. So, nucleoporins are important in RNA transport. True, nucleoporins are important because nucleoporins are only going to, these are the nucleoporins are only the pores that actually helps in the transport. Second is different RNA species follow different pathways of RNA transport export. Is it true or false? So we have already discussed a wide variety, I mean, the range. There are different transport mechanisms for tRNA, miRNA, siRNA, snRNA. So what should be the answer of this question? Different RNA species follow different pathways of RNA export. True or false? I'll give you 2 minutes. Right now it is 1:23. I will switch the screen in 1:26. Okay? I mean 1:25. 1 more minute left. Okay, now I'm switching the screen. So yes, the answer is true. We have already studied that different species of RNAs have different mechanisms of uh, transport. Next is exosome degrade RNA from one end. True or false? Exosomes degrade RNA from one end. Right now it is 1:26. I will switch at 1:28. Okay. Think and answer. 1 more minute left. Okay, now I'm switching the screen. So the correct answer is true. And what is the explanation? So first of all, what are exosomes? Exosomes are nothing but multi-protein complexes that degrade RNA from one end. Okay, this is the definition. Primarily functioning as 3' to 5' exonucleases. Okay, so these are multi-protein complexes, exosomes. These are multi-protein complexes and that they degrade RNA from one end. And they play a key role in mRNA turnover. They also are important in rRNA processing, quality control of RNA, and they degrade defective or unnecessary or excess RNA molecules in both the nucleus as well as in the cytoplasm by progressively removing nucleotides from the 3' end. So what they do is that first of all they work both in the nucleoplasm and in the the nucleus and in the cytoplasm both. Okay, so they work in the nucleus also and they work in the cytoplasm also. And basically they what do they do is that they progressively remove the nucleotides from the 3' end. Okay, what they do is they progressively remove the nucleotide from the 3'. So the question was, the exosomes degrade RNA from one end. Yes, it is true. They degrade RNA from one end only, and mostly it is the 3' end from in which they act. Mostly it is the 3' end. Okay, and they are important or they play important roles in mRNA turnover, in rRNA processing, in quality control of RNA. Next, we come to the next question, which is probably the last question for this class, which is de-adenylation do not favor RNA stability. True or false? Right now it is 1:31. I'll switch the screen in 2 minutes. Okay, I'll come in. Just read the question. Okay, now I'm switching the screen. So the question is, de-adenylation do not favor RNA stability, and whether it is true or false. So it is true. How it is true? Let us see and try to understand this question. First of all, we should know what is de-adenylation. So de-adenylation refers to the removal of poly A tail from the mRNA, and it ultimately leads to instability. So from the meaning itself, we get to know that does de-adenylation favor RNA stability or not. Okay. So de-adenylation, it refers to the removal of the poly A tail from the mRNA. So now we have studied, this is the 5' end and this is the 3' end. You have poly A tail here. And these 5' capping and 3' poly A tailing are actually stabilizing the mRNA. Okay. These two things actually stabilizes the mRNA, and it helps in increasing the stability of RNA. So if you have de-adenylation, which means the removal of the poly A tail from the 3' end. So when the 3' end poly A tail is getting removed, it is increasing stability or it will decrease stability. Of course, it will decrease stability. है ना? So the correct answer would be true. Because this poly A tail, it stabilizes the mRNA and it protects it from the exonuclease activity. And de-adenylation is the first step in the mRNA decay, which makes the mRNA more susceptible to exonucleases like exosomes or XNRN1. So since de-adenylation reduces RNA stability, the statement is true. So I hope this is clear to everybody. Just a minute. So now very quickly we'll try to summarize what we have learned today. So we started with the mechanisms of intron gain. So how intron is gained? So there are two mechanisms for it. First is your intron transposition and second one is your intron insertion. Intron transposition, what happens? The intron which is removed from by splicing from one gene gets attached to the other gene, and transposome insertion means the transposome is it's inserted and then it becomes after insertion it is converted into a spliceosomal intron. And then we studied about what is the meaning of homolog and homologous, orthology and paralogy. So homologous genes are the ones which have a common ancestry, and these homologous genes can belong to two different classes. First, they either these homologous genes can be orthologs or these homologous genes can be paralogs. Orthologous are the ones which are separated by speciation event, and paralogous are the ones which are separated by the gene duplication events. Okay. Then we come to the regulation of the post-translational protein modifications. So how the post-translation protein modification which regulates, these are the phosphorylation, acetylation, hydroxylation, ubiquitination. And then we studied the pathologies which can help, just a minute. Yeah. So the pathology, so the pathologies, can happen with the help of mutations which affects the splicing factors or the patterns of pre-mRNA. So splicing factors like spinal muscular atrophy or retinitis pigmentosa, myotonic dystrophy, or here it can be beta thalassemia, cystic fibrosis, Fraser syndrome, and all these things. Then come to the RNA helicase, how RNA helicase and what are the different functions of RNA helicase. We saw that they are involved in pre-mRNA splicing, RNA maturation, RNA export and translation, and how they are involved that we have studied. Then we have studied about the nuclear core complex, how this RAN, GTP and FGN cargo, everything happens. We have dealt. Then we started the different transport of different, and then we have studied about the miRNA biogenesis and function, how miRNA is formed and how it is exported through the cytoplasm. This also we have studied. And then the various different export of snRNA, export of snRNA, export of mRNA, export of rRNA, how the export of different species of RNA happens, that is there. Then how it is linked to diseases, like how, if what happens if the export, see, first we have studied the disease which happens because of splicing defects. Okay, first we have studied about the splicing defect genes, and then here are the disease which are because of disease I mean changes or problems in the export. So disease can happen because of splicing defects, and disease can happen because of this of any problem in the export also. Okay, so you see here, the disease name is osteogenesis imperfecta, and this disease happens because FMRP gene gets destabilizes. And why this FMRP gene gets destabilizes? Because it interacts with NXF1 homolog, which is NXF2, and it gets destabilizes. And when it gets destabilizes, this is a disease which is osteogenesis imperfecta, which is associated with aberrant mRNA transport export. Okay, and then we started with RNA decay, that how RNA decay happens because the two stability determinants, one is at the 5' end and another one is at the 3' end. These two stability determinants, if they are affected, if there is any problem, then what happens? Your RNA decay happens. If there is any degradation of the 5' end, or if there is any degradation at the 3' end, then your RNA decay can happen. So, and then we have studied about the RNA quality control, that quality control happens at each and every step, from transcription to capping, splicing, polyadenylation, export to cytoplasm, transport, translation, and ultimately, this is the life cycle of the RNA. This is the lifespan, and in this whole degradation happens. So this is all what, and then we have dealt with the questions. So with this, we complete the uh, week 7's live interaction session, and for the next week we'll meet at Tuesday only from 7 to 9. We'll meet in Tuesday, and we'll deal with week 8 section. So I hope this all is clear to you, and I hope all this is clear to you. Whatever we have studied up till now, this all is clear to you. So now we'll just stop the class now, and we'll meet next week. So bye. Okay, so we'll stop the session now.